Okay, good morning again. This is lecture 2, and welcome back to uh, the Bio331. And as a quick reminder, I know you all want to do well in the class, want to go to medical school and go to AP school. And one of the key is to pre-read, prepare yourself for the lectures. And the lecture goes by really fast and covers a lot of material. And so it's better that you understand what's being covered, and if you have questions specifically, then during the lecture time you can raise it. I can pause and and then discuss further a bit more. And again, and I would encourage everyone simply just jump in, and if you have a question, just jump in as if you're in the classroom. In the classroom, you just chime in right away and say, okay, what is this and what is that, right? So that's that's a little bit common, and encourage you to prepare the class. So this lecture covers the cell structure, function, and molecular composition. And so these are the 5 topics that we will be talking about today: cell morphology, microscope, cell membrane, cell wall, motility, and inclusion body. And some we will discuss more than the others. So the cell morphology, the first topic we want to know is cell morphology and microscopes. And I know you have already spent a whole lab time on microscope and looking at the microbes. And so you would have seen already the 3 basic types of microbes: the calcite, the rod, and curved. Okay. And then microbes have lots of exceptions, and there are lots of different other shapes, uncommon shapes. Okay. And so you have spirochetes, and then you have microbe with stalk or hypha, and so all these have specific functions. For example, the stalk, and this is a bacteria that they use this stalk to attach themselves to something. So like say on the side of the stream, maybe attach themselves to the grass. So then the water flows through, the nutrient comes by, and they don't even have to do anything. The nutrient just comes to them, right? And then you have filamentous, and filamentous is also pretty common in rivers and streams, and they attach to one end, and then again nutrient flows by, and so it's very convenient for them. And so here are some real examples, real life examples. And so this is bacillus. Okay, and you will have to be able to recognize some of the basic bacteria. So this, for example, this is a chain of bacillus, and the bright spots, this is under regular light microscope, the endospores, and turn out to be white. Okay, this is an artifact, right? We know endosport is very dense, and but it should block light, right? And this is Streptococcus, forms a long chain. And so often these are 2 bacteria actually. And so if you look at this, and this is a little wrinkle here, goes back in, and there are actually 2 dividing bacteria. And these 2 are just divided, right? And so they're far apart. And this is an example of filamentous bacteria. In terms of the size, so those are the shapes, right? Basic shapes. But in terms of size, bacteria we already know E. coli, right? And you should be able to recite the number 1 by 3 micrometers when you asleep, okay? And so these are, this is Streptococcus, and these are some basic bacteria size that you should know as a key reference. So people will say, okay, how small is small? Well, small is E. coli is 1 by 3. Now, your hair is very thin, right? How thin is your hair, right? It's a 20 to 30 microns, or 200 microns, or the hairs, everyone's hair is slightly different. So you need to have some sort of reference. Then you have you have other bacteria. So this is cyanobacteria. Look at the size, and this is draw in proportion. So it's 8 by 50 micron. Now 50 micron is big enough for you to see invisible, for you to see. So if this bacteria is when you put it in the microscope that Leeuwenhoek was created, put it on the needle. You don't even need to go through the lens. You can see it directly in your eye. So then that creates a question of, well, they're all bacteria. Why the size varies so much? Is there any advantage being big or being small? Okay, so if you look at the volume to the surface ratio, so if you create the ratio between the surface and the volume, right? And the smaller one actually gives you more surface than for the volume, right? So basically, if you imagine if you combine these little drop into larger drops, yeah, then the surface area will actually reduce per volume, right? So this what this means is that the bacteria requires the import, right? Like the mouth. The more surface area you have, the more mouth you can put, or transporters that you can put on the surface. What that means is that actually there is an advantage for being small, because the smaller the volume, and you get more transporters per volume on the surface. So then becomes more efficient in taking up nutrients. So if we go back to this example, now how does cyanobacteria get energy? Anyone want to chime in? Anyone want to guess?
Through the is it the same way like plants, right?
Yeah, look at the color, right? It does photosynthesis, right?
Yeah.
Now, what about E. coli? Does E. coli do photosynthesis? Excellent. Yeah, guys getting it. No. Okay, so E. coli will have to get their energy based on what they can eat, right? So cyanobacteria doesn't care. It doesn't need the light. It doesn't need to mouth to get the food, and light just penetrates right through the membrane. So in a way, you can think about this as, well, cyanobacteria doesn't need to eat for energy, and E. coli need to eat for energy. So E. coli cares more about having more mouth per volume, and being more efficient in taking up energy or food, right? And so that's an explanation that the difference between the volume and the size. Okay. So that's one explanation for the size. Now, micros are, regardless, majority are very, very small. So you have to use a microscope. That's what define our field, right? Microbiology is the study of microbes. What's a microbe? Well, microbe is something you can see. You cannot see without the microscope, right? Um, so microscope is the main tool, and if you haven't noticed, the in-person lab that we set up is the really one of the things that we need you to really get a handle on is the use of microscope. Okay. Now, if you complete this class and go to the next class and you still don't know how to use microscope, and people will, again, you're going to lose a lot of credibility, okay? So it is very important. Now, one of the key things about microscope is the resolution. How resolution is defined by you've learned that in the lab, right? And how the distance between two dots that you can resolve. And and we have three different main types of microscopes. One is a light microscope. Okay, the light microscope, optical light microscope. The main resolution is 0.2 microns. Okay, but that's regular light microscope. But we've come to rely on lots of improvements over the years to improve the resolution visible, right? And then the other is electron microscope, and that is high resolution because resolution depends on the wavelength of the light. The electron beams that we generate has no wavelength; it's just straight. And so that gives you the highest resolution. And transmission, it's transmitted microscope that gives you even higher resolution than scanning EM. But scanning EM does provide an interesting advantage because the image it generates is a three-dimensional because it's based on the bouncing. So I'll talk more about these two classes of microscope a bit more later. And hold on 1 second. Okay, sorry about that. So let's first go through the light microscope, and you've already seen the light microscope in the lab. This is your eye, and right, this is structure, and this is the eyepiece, and this is objective lens, and this is the sample. And this is the light path, right? And the light gets condensed and then comes through. Now, this is the basic type of microscope and determines the resolution. And so, but it has its limit, the quality of the light. And now for last time, there was a question about the curvature and the size of the lens. When you were in the lab, don't forget to look under the objective lens to see how big the glass is, right? And you will see that 100x the oil lens that little hole, pinhole, is tiny. That's where the lens is. Okay. Okay. Now, to improve the light microscope, the first thing people did was to use staining. Well, because the stain, if the bacteria is stained, then that's different from the environment. Because your ability to see things on the slide depends on the contrasts, right, between the sample and its environment. Okay, and so here are different stainings, and you will get to learn how to do gram staining. And let me just lay the question here: is gram staining an absolute reliable technique, and you can always tell the difference between gram positive and gram negative? Can someone, you don't need to answer this question now, but I'm just gonna lay a question now for you to think about it. When you do gram staining, could it be a mistake if someone tells you, "Oh, I've seen this bacteria is gram positive"? Could that person be wrong? Okay, you don't have to answer this question for now. There's a question from Catherine. Go ahead, Catherine. Don't wait for me.
Oh, no, I was gonna answer it.
Don't answer it. I want to keep everyone suspended so that your lab professor will go through this.
Okay.
I wanted to see how many of you are awake because I don't see your pictures.
Professor, can you repeat the question?
The question was, when you do gram staining, you use gram staining to see a bacteria, whether it's positive or negative. Could the result be wrong? And is it possible to stain a gram positive bacteria after you do the staining and come out to be gram negative, or vice versa, a gram negative bacteria appear to be gram positive when you do the gram staining? We don't have time to discuss, and I just want to keep everyone in suspense. Ask your lab professor. Okay, now. So one way is to increase. We'll come back to this staining and sample preparation. So there are two sides to these things, right? The sample preparation and improving the microscope. So now I'm going to discuss what are the things people have done to improve the microscope itself. Okay. Let me close my window. A bit noisy. So this is a basic light microscope, and it's similar to the ones that you've been using. One of the first things people have done to improve it, the microscope, is called dark field microscope. Okay, so one thing that what they did was to put a block in the middle of the condensing light. Okay, in the lab, you basically just put a bottle cap, a black bottle cap, in the middle. And so what does that do? Well, it creates. So this is. Everyone can see my cursors, right? And my hand is moving. Okay. So this is a regular.
No, we can't see it.
No?
Some of my other professors were having trouble using the cursor and pointer in Blackboard. Dr. Kennelly last night could not use it as well. There may be an issue.
Oh, yeah. That's really strange because I can see, I turn on the hand and it's supposed to let me point out things. Okay, so hold on 1 second.
Professor, use the pen.
Yeah, okay, I agree. Use the pen. That would be better. Use the pen. Okay. What did change? Okay. Okay. Sorry. Okay. Let me do this. Okay. So this is the light microscope. Okay. And you see the background is grayish. The bacteria is that. But then this is the dark field microscope. Okay. So this is the dark field microscope. Now you see the difference between the background being this is black and this is grayish. So what happened is that if you block the light from the middle of your field, and then the light will actually come from the side. Okay, so you see here is exactly what's going on here. The beam, instead of going through the middle, it goes from the side, right? And the side then shines the light on the object, on the bacteria. And so the light actually comes from the side and then bounce off on a black background. And then it becomes more, it's easier to see, right? So a way you can think about this is that if you have a plastic, very thin plastic white string, transparent string, it's almost invisible. Then in order to see it better, if you put it on a piece of black paper, would it be more visible, right? It will be more visible. And another example to think about this is, well, if you go to a movie and if someone is, no one smokes, but if you go to a movie and the light shines through, you can actually see dusts that can come going through the beams, right? Those dust particles normally are invisible. And if we were to be in person, in an in-person lecture, I often point out to the projector lens, the space very, very close to projector lens. If you focus your attention there, you can actually see things, dusts floating around, which is not visible. You know, you think the air is very clean, but we constantly shed, release skin particles, and that goes into the air. Okay. And so that's the first improvement, and it's really effective. And what's it's useful, most useful, is that if you get water sample, and if you have these single cellular microbes that have a thin membrane, let's say protozoa or anything, and if you shine light through, use dark field microscope, and that makes it stands out so much more. So that's the first improvement. The next improvement is called the phase contrast. Now the phase contrast will take me 2 hours to explain, but in general, what happens is that they use special lens to separate the light into two beams, and one so that one goes faster than the other, and then they will combine the light in your eyepiece. So you see the net result is that the background. So this background here is not black, but it's darker than gray. So imagine this is basically they use the light to cancel each other out a little bit. Okay, they canceled about half of the wavelength. So now it's a little bit that's dim. But when that slow light passes through the bacteria, and then slows down further, another quarter, and then the two peaks Let's see if I can do that. Okay, right? That's one wavelength, right? And if you have another wavelength, can I try a different color? Blue. So if the wavelength is a half, terrible, I'm not a physicist, but if the wavelength is like half wavelength slow, and so you see this one is exactly half, and so when they are half wavelengths in difference, they cancel each other out, and then gives you this black spot. And so, so what the advantage, it's difficult, I know it's difficult to follow, especially with my terrible drawings. But the key here is that the phase contrasts further stress or further expand the difference between the bacteria, the contrast between bacteria and its environment. And so that the bacteria becomes black, and what's the advantage of that is the interior structure is much more visible now. It's in higher resolution. And if you compare the phase contrast with the dark field makes things stand out. But we all also know when light is glowing, reflecting, it's difficult to pin down more precisely measurement. And also the interior structure is not as clear. So this is the same image that viewed in three different settings, right? And so you can see the internal structure much more clear in the phase contrast. Okay, I'll pause for a second because these are the three basic modifications. Does anyone have a question? Okay, so those modifications have been done like 50 years ago. In the last 20 years, or even more than that, last few years, and we have, or people have been using fluorescent microscope as a way of improving. Okay, so what's drawn here is the light microscope, and now the red here is fluorescent light source. So this is like the same light that's coming out of the projector, right? The video projector, you see it's very, very strong, right? And that's the same mercury lamp that we use to generate fluorescent light source. However, the fluorescent light is white, right? The mercury lamp that comes out is white. And so what you do then is you put a filter in front of it. And so then you can select different wavelength. So you can select green, blue, whatever filter you put through. And then the light comes through the microscope and then shine onto the sample, okay? Now, the reason you do that is, one, because the light is shining onto the sample, not towards you, then anything, if the light did not hit anything, right, and then that area will be completely dark. Now, obviously, the entire setup needs to be, the fluorescent microscope needs to be used in a dark room, like no light. And so now the background is black, and then because the fluorescent, there are material that give you fluorescence color, right? Well-known fluorescence, that you shine a light and then they give you a different light comes back, right? Fluorescent light and neon light, almost like, right? And so we have materials. We can then couple the material to say a antibody or any material can bind to specific structure. So for example, we're looking at the actin filaments here, and then you shine the light to it, and then you actually get the green fluorescent light. And these fluorescent light are very weak, and so then you will need to be on a black background, so these weak fluorescent light can come out. And the advantage is that you can use different stains. In this case, two different stains are being used. One is stained actin filaments, then the other is staining DNA. So now you can actually visualize the actin filaments and the relative position of the nucleus. Right? And so this is a more recent improvement. Okay. So that's the fluorescent microscope. And now you understand when we lecture in the regular in-person lab, you will see a professor using a laser pointer, right? And pointing out, the laser light will project a red spot on the screen. And the point I want to make is a laser, that's a vivid example of laser, is actually stronger than the fluorescent light, that the mercury lamp that we are using for projecting the image, right? Okay, so because laser focuses, and so now the earlier I said the problem with fluorescent light is the fluorescent light itself is this green light is very, very weak. And so what you then do is if you replace the fluorescent light source with a laser light, and with a laser light, then that's even stronger, right? Now, so the laser then, what does laser do is it's a spot. So then what happens is if you use laser, you basically focus all the light into one spot. Then obviously that makes it difficult, right? So it'll be one spot here. Well, you don't see anything. So in order to use the laser, then what they call is they use a laser to scan. So that laser, one spot goes from here, here, and here. Now, if that laser light hit this spot, the fluorescent light will be orange. Why? Well, because it's hitting the nucleus stained fluorescence, and your computer then records that spot. And then when the laser comes here,
Professor, sorry, pointer.
Yeah, no, okay, sorry. Yeah, there's no pointer. Okay. Okay, let me try again with my pen. Okay, so the point is that it goes through, like if this is my laser, okay, it just goes through, and then it comes down and hit, and then when it comes here, it hit the nucleus, and then gives you fluorescent, and then it goes down and goes down here, and then start hitting here. Now, if the laser hits here, then it hits the actin filaments, right? Did someone have a question? Go ahead.
Oh yeah, I couldn't see it, but now I can see it. So.
Oh, okay, yeah. So this is so the point is that when you use a laser, it actually scans through the whole field. Okay. All right. Now I'm dramatic, all right, and it wouldn't skip, right? Because mechanically, that's just a dense scanning. So what does it do then? Well, okay, use the laser, and the laser. There's a lot of mechanics involved, and so here is a laser scanning head, and look at this one. It's tiny, right? Well, it's not that tiny, but it's small. So what's inside is actually very good. Let me just explain and say, oh my God, this is the most complex graph we'll have. Never see anything this complex. Now, when you see a graph this complex, you're going to need to grab a handle arm and say, okay, how do I understand this? Okay, so you go to the light that comes in. Okay, the light hit a mirror, goes down here. Wait, hold on. I need that pen. Okay, so the light comes here, hit the mirror, goes down here, hit another mirror, and comes here and hit this mirror. What does this mirror do? It says y-axis. And then it goes from here to hit this mirror, right? Where's my, and then this mirror, and then this mirror. And what does this mirror do? It controls y-x-axis. Figure the previously what I was trying to say, okay, it's a scanning, right? It goes, this is x, y, x, right? So this then I can do, I'm sorry, but the point is that you can do scanning, right? With these light, this mirror controls the light spot. And then the fluorescent light, when it goes into the microscope, hit the sample, and the green light comes back and goes, you mean the light is very, very fast, right? Travels very fast. It's instantaneous. And so the fluorescent light will come back instantaneously. Let me change the color. Oh, I want green. Okay, so the green light, right, comes back here. And here, back here, here. And then it goes through the mirror. And this mirror is half transparent, and you can it's manufactured so that it allows certain wavelengths to go through, and then comes here. Okay, and shows the mirror, and then comes back here. Now here's the interesting part. You have three photo multiplier, meaning like the eye, and if a photon hits it, it will generate a pulse, electricity. It will register. And so let's say if this is a green, so this is green, and so then the green light will come here and goes through. But if the light comes here and this future is set to red, then it stops. And you can go down here and nothing happens. So green light will only be registered with one by one of the multiplier photo sensor. So now you have three sensors sensing three different light source, right? And so the confocal then microscope can separate into three different colors. And if you label three different biomolecules with three different label, then you can see where they are relative position. So green and blue, that if you mix green and blue, that gives you, or red, that gives you yellow, right? The green and red, that's yellow. And then you added a little bit blue, and so this is, so now you know these two are co-localized, right? Depends on how, if you keep zooming in, you can actually see how closely they are, the two molecules are aligned to each other. And so this is the fluorescence and the confocal scanning EM. And so obviously fluorescence are very, very expensive. Confocal is even more expensive. So when we talk about money, things are really very expensive. And so guess what? We do have a confocal microscope. And does anyone want to guess how expensive our confocal microscope is? Like in the hundreds. Okay, so yeah, well, when you talk about numbers, you have to give it well, 150, that's you have to double that. So the one that we have is actually 350,000. That's about years ago. Then nowadays, you can confocal microscope can easily get to 1 million. It's all the things. Why is it so expensive? Well, it's very mechanical and very electronical. Dealing with light, and the weak light is very, very challenging. And they can even do, the latest is that they can do single cell labeling, because they can control the light. They can wipe out. I'm being carried away. Sorry. Just say that they can see proteins using fluorescent light. Okay, so that's where we are, and the regular microscope that you're working with in the lab is about 3,000. A phase contrast microscope that's sitting in the front is about 10,000, and a regular research grade fluorescent microscope will run you somewhere between 20 to 50,000. And a confocal, and a fancier fluorescent microscope will run you about 100,000, and a confocal will run you about 300 to 1 million. Okay. Now, so those are the improvement with the light microscope, and then we go to electron microscope. There are two different types of electron microscope. One is a transmission, one is scanning. The key difference is the electron beams. Okay, that comes through here. You see these red arrows indicating the electron beams, and the sample is here. And so you see in this case is transmission, meaning the electron beams that really goes through, and then hit the sensor. I'm sorry, this should be straight line. I just kept. The difference between this transmission and then scanning is scanning the electron beams hitting the sample, and you collect the reflection. Now, it is very, very, very important you understand we, or the living organisms, all have carbohydrate molecules, right? We're not dense. We do not reflect electron beams. And so if you want to see any sample, living sample, you have to cover it up with heavy metal, mostly gold. So you decode the sample, you put a layer of gold on a sample in order to see it. And so in this case, this is a transmission electron microscope sample or graph, okay, a picture. And this is generated using scanning. Remember I said the scanning gives you a three-dimensional image? See, this is a scanning three-dimensional image. And this is transmission. It's like a cross cut, okay? It's like a sectioning. And then they actually do. So what we do is we actually embed the bacteria in a plastic, and we cut them into a very, very thin section. And then then allow the electron beams to go through, and then you see the difference. You do stain the sample. You have to stain them. Okay, so that's a quick summary. Summary of the modification of the light microscope, electron microscope. And there's one thing I didn't comment on early, is called the atomic force microscope. It's super high resolution, and you can see living samples. So you see electron microscope and confocal, they are all from the They're all looking at the stained sample, and the samples or living things are dead. They have to be killed. Now, for what I forget is that you have questions about materials from which chapter. And one way to look at this is to look at the figure legend. Okay, so figure legends are all labeled. See, this goes to chapter 4 and figure 15. And so this gives you a handle on where you find the material, find the ones that you'll read immediately. So let me go back to the atomic force microscope, and this is really funny. And now, if you want to, I'm just gonna stop. Some of you might have done it, but if you haven't, if I ask you to generate an image of a penny, what would you do? Do you draw an image of penny? It's a perfect time for a 1 minute break. How do you draw a beautiful image of penny? How do you generate them? Let's say draw. Some of you must have done it.
Professor, you could put a paper over it and use a pencil to lightly trace over.
Okay, you say trace over, you mean you use a pencil to go draw across the penny, right? You end up,
like shading it in.
Excellent. That's exactly what a atomic force microscope is doing. So if you use a thick pencil versus a very, very thin pencil, and the image that you generate will be, a thin pencil will be much more higher resolution than the a thick image, right? Let's say you use a Sharpie, you won't generate something. You will generate a blob, right? Versus if you use a very sharp pencil, you will generate a very high resolution image. That's precisely what the atomic force microscope does. So it creates a very, very thin needle, and then it drags across the sample, and if it goes, it dips or ups and downs, and as it goes through, and you record the image, the image is recorded, okay, and so the advantage of that is give you high resolution. It depends on how thin the needle is, and then it's a living sample. And the best thing to do is to look at the cell wall. Say if you have a tomato skin, right? You lay down the tomato and then you drag you through. You can actually see cellular structures of tomato skin cell. This is just high resolution. Okay, and good. All right. So those are microscope and different type of modification. And so next, I wanted to comment on the basic structures. And now these are the things that you should have already learned a little bit. And so half of these, the segment is review, and well, I think mostly is review because you already know most of it in the intro bio or genetics. But let's go through it. So the cell membrane, we'll go through the structure and the function. So the cell membrane is the basic structure that keeps a cell a living cell. All right, because this is the structure of cell membrane. So it's made of glycerol phosphate, right? It's a bilayer membrane. So it has the hydrophobic region in the middle and hydrophilic region on both sides. And so the basic molecular structure of the membrane, it's like this, right? This is the glycerol molecule, right? 3 carbons, it has 2 fatty acid chain, and then it has a phosphate here. And it has some phosphates, sometimes have modifications. Now, this is phospholipid. And what's the difference between phospholipid and regular lipid? Regular lipid is what? Oil, right? Fat. And the difference is the regular lipid or the oil has 3 fatty acid chain, okay, instead of 2. So the oil then has the hydrophobic region because fatty acid likes to stay with fatty acid, right? And that's what that is. And so you can actually stain, use different dye to stain the hydrophilic region, the phosphate. And then if you do that, and then you can see, you can actually visualize the 2 membranes. Now the question here is, what kind of microscope is used to see the membrane structure? Anyone want to chime in? Light microscope, fluorescent microscope, atomic force microscope, transmission electron microscope, scanning electron microscope. Those are your choices, right? Think about the resolution, okay? What's the question? The question was, what kind of microscope was used to see or to generate this image of a membrane? Yes, Sean, thank you very much. That's excellent. It's transmission electron microscope, or TEM. Very good, thank you. Well, the membrane, so that's the basic structure of the membrane. Now, membrane is not empty. The membrane is actually very busy. The phospholipid is actually filled with all the membrane, a lot of membrane proteins. So this is the outside, and this is the inside. Okay, inside meaning inside the membrane, inside the cell. And remember earlier we were discussing the size versus the volume. Well, we said, well, if the volume to size ratio is high, you get to put more proteins. You get to have more mouth, meaning the transporters, and these are transporters. And so on the membrane, you have lots of proteins, and so they can actually get across. We'll come back to these different types of transporters in a second. But for now, I'd like to highlight a difference between these 2 membranes. Can you see the difference between the 2? Look at here.
Yeah.
Look at, right? And so, so the bacteria has a lipid bilayer, and Archaea, this is Archaea, has lipid monolayer, 1 layer. Okay, so the membrane, the Archaea, remember we have bacteria and we have Archaea. For Archaea, the membrane is different. It has monolayer. One of the ways you think about this, how do you keep track of who's monolayer, who's bilayer? So monolayer, obviously, it's stronger than bilayer, right? Because you can't separate them. Versus bilayer, you've given enough force, you can disrupt them. Well, and so monolayer is stable, and Archaea tend to live in extreme environment, very hot or very salty. So they need their membrane to be much more stable. And so the monolayer will provide more stability to the membrane. So now your conclusion is, aha, the monolayer is linked to Archaea. Well, because Archaea lives in extremely hot environment. Okay, good. All right. So that's the membrane structure. Now look at the function. What's the function? Why do we have to have membrane? Well, membrane is a permeability barrier. It prevents things from leakage. So basically, it's a barrier. Nothing goes through. There's one molecule can go through the membrane very easily. That is water. So the water actually goes through the membrane just with no barrier. Okay, just goes through easily. There's consequences to that. And then every other molecule needs to go through the membrane with some sort of transporter. If there's no transporter, it can go through, but very, very inefficiently. And so proteins on the membrane, a lot of them serve as a transporter. Okay, and oops. Wait. Sorry. What did I do? Okay. And then you have protons. And the proton doesn't go through the membrane. And the proton, which will become very, very important, okay? The proton is a source of energy. Remember hydropower. What's the hydropower? Like you build a dam and then water accumulates and becomes very high, and then you allow the water to go through and to drive to push a rotor, and then generates the energy, right? And so or like if you think about earlier, people use sawmill. They use stream water to turn the wheel, and then that's source of energy, right? So the proton, we'll come back to this very often, is that almost all living cell generates proton, and then the proton gradient is used to generate ATP. We'll come back to this later. So now, before we move on, is that if you lose the integrity of the membrane, so if you have a hole, then you will not have this proton gradient, and the good stuff will leak out, well, like the energy molecule, the ATP, the protease, or whatever you have inside the cell will leak out, and then you don't have a living cell. So the membrane integrity is very important. Now let's look at the transport. And so the transport, let me just warn you, can be very confusing, and they can be classified based on many different criteria. Okay, a transport. Okay, so, well, you can classify, think about our cars or automobiles or cars or trucks, right? You can classify our cars or vehicles based on the size. So then you have big truck, school bus, maybe little your own cars, right? And then you can classify based on what kind of things they're driving, provide the energy. You can have electric cars, you can have or electric vehicles, or you can have diesel vehicles, you can have gasoline vehicles, right? So when you think about the transport, you need to be very clear on what are they asking. And then you can see what kind of system they are classifying, because a transporter can be classified in many different, could be called different things. Right? Okay, so here's based on it's transporting system. So this is a simple transporter. It just goes in, right? Takes one molecule, goes in, and uses proton gradient to drive through, and you can have group translocation. So meaning that when this molecule comes in and it adds a phosphate group, or you can have an ABC system that actually sends out a scout. Another protein goes out and binds the molecule and brings back to here and transports. And this is called the ABC system. Okay. What's common about this? Well, it's all require energy to transport. So it requires proton gradient to push us through this molecule, requires a phosphate to put it on this molecule, requires phosphate to bring this in. Okay, so this is an example of three membrane transport system. And if you look at a transport versus non-transport system, you can look at how efficient the molecule actually gets through the membrane. So this is this axis, the X is the external concentrations of solute. So here is low and here's high. This is high. Oh, sorry. And this is, oh, I have to type it again. This is low. So this is about concentration, right? And then if you look at the rate of solute entry, they measure by how fast it comes into the cell. If this is very slow, right? But it's concentration dependent. The higher the concentration, the more you get into, but very slow. And so this is a typical transport pattern for diffusion. And this is a typical transport pattern for carrier-based, like have a transporter. And so if you have, so as you increase the concentration, if the as your concentration increases, and this goes up quickly, but then it saturates. Why does it saturate? Well, the numbers of your carrier on the surface is fixed, right? Right. So when it's here, it's very efficient. Okay. If you have 10 transporters across the membrane, and so when the solute comes in, it gets transported in immediately. But when you get to a certain higher level and you saturate all 10 transporters, well, then the rest had to wait. Then the rest becomes almost like a diffusion, right? And so by looking at how fast a solute can get across membrane, you can easily determine whether it comes through by diffusion. Now, there's another way of, there's different ways of looking at transporter. This is I like to highlight, this is the outside, and this is the inside. And look at this one, it's a uniporter. Why? Well, because it comes in just by itself. So that's why it's a uniporter. And then this is an antiporter. Why is antiporter? Well, because this is substrate that comes in, but it's coupled with something else that's going out. So it's in opposite direction. And this is a symporter. So these things comes in together. So this is symporter. All right. So this is another way of separating out the transporter system. Here is something that real life example. The sodium. Well, sodium is what? This is. Anyone want to try? This is symport, uniport, or antiport. This is an antiport, right? Because sodium goes no, because symport is same direction. Antiport is the opposite direction. So yes, Catherine, you're right. And so the antiport so relies on the proton gradient to bring out the the sodium. Okay, so now we all, when you sweat, what happens? Your sodium comes out, right? So now you can say, when I sweat, I'm using my proton to pump out sodium. And so here is what happened? Here is a symport. Phosphate is a symport, and potassium is a uniport. And then, so these are things, but then you have enzyme-based. This, we'll come back to this later, but this is Lac Permease. It is very, very important that you understand. A permease is an enzyme on the membrane that specifically brings in a particular molecule, and it tells you in the front. And it's a Lac permease, then this permease brings in lactose only. It doesn't bring in glucose. Okay. And then you have sulfate symport, and that brings in sulfate. Well, we do need sulfate for biomolecules. Okay. So these are the well-known transporter systems that we have. And aside from that, you have a glucose uptake system called PTS. And so the glucose, and this is important. This is the outside. So the glucose is outside, and this is inside the cell. This is the inside the cell. And these are the enzymes all coupled together. Okay, so this is also can be called group translocation mechanism. Okay, the glucose, this is how glucose gets taken into the cell, and this is very, very common. Almost all bacteria have this glucose uptake system, and this is the another view of the ATP ABC transport system. Okay, so this has the sample here, and that brings in. Okay, so this is the transporters. This is how that works. So now we're going to need to take a look at the cell wall. So cell wall, so this is membrane, and then outside, so this is membrane, right? Then outside the membrane, you have this cell wall, and we'll go into this in detail. But for now, I want to draw your attention to the difference between gram-positive and gram-negative. So this is where you can see the outside is a peptidoglycan layer, functions as cell wall, and the gram-negative, you have cell wall, but a thin layer of peptidoglycan. But on the outside, you have a outer membrane with all these things, which we'll talk in a second. And so this is gram-negative bacteria, this is gram-positive bacteria. And if you, after the gram staining, this is what you see. The purple is gram-positive and pink is gram-negative. Earlier, I asked a question and keep you in suspense. And could you make a mistake and stain gram-positive become gram-negative with this stain? Okay. But the focus right now is to look at the structure. So this is a gram-positive cell wall, right? You see two layers, and then you look at the gram-negative bacteria, and then you can see here multiple layers. You have cell membrane here, and then you have outer membrane, and then you have peptidoglycan in the middle. And now obviously this graph is generated by transmission electron microscope, and this graph is generated by scanning electron microscope. And so let's look at the gram-positive cell wall for a minute. And so it's somewhat simple. You have cell membrane, and then you have peptidoglycan layer. Okay, and so gram-negative bacteria, and so here you have 3 layers, right? And you see here is 1 layer, another layer, and outer membrane layer. Okay, so the picture of this here really demonstrates how thin the peptidoglycan layer for the gram-negative bacteria, right? Okay, so and the consequences of the cell wall is the SEM picture will be different, and because gram-positive cell wall tend to be peptidoglycan layer tend to be strong, and so we have a straight cell wall, and the gram-negative bacteria tend to wrinkle a little bit because it has the outer membrane when you dry them up. And so what's important, if you do a cross section of the peptidoglycan or the cell wall, you will see that these peptidoglycan sheets, so this is like a sheet, right? And then between the two sheets, it's linked. You see? So there's a linkage between the two sheets, and that's where the things gets, why the cell wall gets so strong. So the way you think about this is, if you were to build a fence, right? I know you're living brown. I know if you have little garden, if you want to build a fence, what you do? You put down like sticks, right? But sticks are individual, and if you want to make it strong, what you do? Well, you use metal wires to link all the sticks together. So now you have a fence, right? Now, if you think about it and say, "Go, I want to make my fence stronger." So what do? I put 10 layers of fence, and if you put 10 layers fence, then you actually combine all 10 layers together by putting the I link one layer with the other, use metal wire, right? And that's exactly what's happening. And so then it becomes really strong. So how is this linkage take place? So this is a detailed look at peptidoglycan. Okay, so the peptidoglycan layer sounds very complex, but the theme here is that the microbes, and you will see bacteria and virus later on, they tend to use one molecule and do many repeats, because it's easier just to make one molecule or 2 molecules and make lots of them. Okay, instead of making lots of different molecules. And so what you see here is this peptidoglycan layer, so many of them, but there are actually only 2 basic molecules. So N-acetylglucosamine is one, and N-acetylmuramic acid. And so this is one unit, and this is another unit. And that's it. And it just goes from G and N repeating. So, right? And then they use side chains to have alanine and glutamic acid, alanine, and mesodiamine malic acid, another molecule. And so they use, so these are what's called a cross bridge, and different microbes uses different. I'm sorry, the side chain. And so here is how gram-negative. And so here's gram-negative bacteria, and they use direct link between these two amino acids and to form this cross-linking between the two sheets. And the gram-positive bacteria, like Staphylococcus aureus, uses a glycine interbridge to bring the two sheets together. Okay. And so that's the difference. But they do the side chains that they have repeating side chains. So you see, the repeating is the same. And they use these side chains to stick them together. Okay, just like the Scotch tape. Now, in addition to this cross bridge, they also, the gram-positive bacteria has one additional molecule to stabilize the interaction, right? And so here is the chloric acid. So there are two types, and it's lipo-teichoic acid here, lipo. And why is it lipo? It's because here it's embed one end to the lipid membrane, and then the other is wall associated. And so this, then you see, it only embeds in here. It's crossing different peptidoglycan layer. And so the teichoic acid is a special molecule, and this is a teichoic acid molecule, and with the phosphate and with side chains and sugars, and so that they can cross the link easily. So to visualize the effect of the cell wall is very, very important. The cell wall is really keeping the integrity of the cell. Remember, cell is a lipid layer, right? The lipid layer is actually pretty fragile. So how do you know? And if you look at here, if you put a bacteria in isotonic solution and then you use enzyme to remove the cell wall, and so what will then happen is the cell will actually flow out of the cell wall and become a protoplast. And so here you only have membrane. And isotonic solution means what? The salt concentration outside and inside is the same. Remember earlier we said water is the molecule that can go across the membrane. And water tend to go from low salt concentration to high salt concentration, so that the salt concentration will be equal. Say, this is really difficult to keep track. Which direction the water is going? Okay. The way you think about this is the water will go in a direction so that the salt concentration across the membrane will be equal. That's the net result. So the water will flow in the direction so that the salt concentration on both sides of membrane will be equal. So if you remember this principle, then you're not afraid. You can analyze, okay, so in this case, it's isotonic. So meaning the salt outside and inside is, well, if it's already the same, does water moving around? There's no need for water to move around, right? Because they're already equal. But in a low solute solution, that means the salt concentration is higher inside than the outside. So like a rainwater, right? So think about rainwater. Is rainwater salty? It's not. Okay. So now if you put a bacteria without a cell wall or degraded cell wall into a low solute solution, then its higher concentration is salt here, and then water will enter to the high concentration. Why? Remember the principle, the salt concentration needs to be same across the membrane, right? And the water will flow. So then if you have one side high concentration salt, low concentration salt, the water will go from low concentration to the high concentration. So now in this case, the water goes into the cell, and then the cell bursts, right? Because it can't take. Just like if you put air into a balloon constantly, and the balloon will eventually blow. But have you ever tried? If you put a balloon inside a cardboard box or whatever container, a solid container, you can put a lot more pressure into it, right? Case in point is if you have bicycles, right? If you have bicycles, if you put air into the bicycle tube, inner tube is like a balloon, it's flexible, and it can expand, and the outside, the hard tire actually provides the structure. And so this is actually, the case tells you how the function of the cell wall and the function of the cell membrane, and how water can go across. Okay, I know this is a bit of complicated. Does anyone have questions? Okay, maybe I explained super clear, or maybe I lost everyone. But anyhow, this part is recorded, and you can go back and read it again. And obviously, you can.
I do have a question.
Yes, please.
So
Yes.
that with a low solute solution it will go inside. What happens when it's opposite? So if it's a high solute solution, and is the water gonna like escape? Is it still lysing if the water escapes?
Yes, that's an excellent question. So what happens if you put these bacteria into a high salt solution? Well, if you put it into higher salt than what's in the bacteria, then water will go from low salt to high salt, meaning the water will come out, right? And when water comes out, and then this bacteria will actually shrink. And then if they shrink, the bacteria has no water, and if the cells get shrinked, like plumes, right? Plumes, right? And the dried fruit, or raisin, right? Think about grape and raisin. And raisin, take out the water and nothing will grow. Raisin can stay for a long time, right? Grape won't. Grape can be fermented, or bacteria can go in. So the water actually, if you take out the water, and that's how the bacteria will stop growing, or living things will not be able to grow. And so preservation, I mean, if you go back, even my grandparents' age, well, when I was young and we didn't have a refrigerator, I was probably 10 or 12 that our family got the first refrigerator, right? But before that, what you do? Well, you put lots of salt. Many cultures use salt, or well, salt is most common because it's cheap, to preserve meat and lots of things, right? Or you put meat in the air, let it dry. What's dry? Dry is taking out water, and you can use salt or air to take out water. And so, I'm sorry, I carried away, but in this case, if the bacteria is in high salt concentration, the water will come out. Good? Okay. Okay, so that, okay, thank you. So the, it is, yes, water activity from yesterday's seminar, exactly. When water activity is determined by how much water. If you remove the water, yes. But that is dependent on your understanding of the membrane. And the understanding of membrane is a barrier so that water can cross, but nothing else can cross. Salt cannot cross. Good? Exactly. Okay, so now we can look at the peptidoglycan. Okay, so we talked about peptidoglycan, the Chloric Acid, and how it's breaking down the effect. And then you have what's called the Pseudo Peptidoglycan, because I keep coming back to this later. The Pseudo Peptidoglycan is different from peptidoglycan, and they don't have repeating units. It's a little bit more complex. And so the net result, as later on you will see, maybe I mentioned earlier, that the Archaea versus bacteria, Archaea, the pseudo-peptidoglycan, Archaea has pseudo-peptidoglycan. They don't have peptidoglycan. And they don't have a neat repeating unit. So their cell wall is irregular shape. Okay, now I want to get to the cell wall, and then I want to get to gram-negative cell wall and highlight the difference. The difference is the outer membrane, and the outer membrane has lipopolysaccharide, and this even gives you the distance, and then the gram-negative cell wall cell outer membrane has this unique molecule. And let me change the color. The green doesn't show up well. Okay, so these called pore ring. So just like a pipe, allow things to go in, because the selective transporters are here. The transporters will be on the inner membrane. The outer membrane is really just a barrier, right? And the key here is that it has lipopolysaccharide, and this is the basic structure of lipopolysaccharide. And yesterday, if you went to the seminar yesterday, there's one bacteria called O157. What's O? This is the O. Because the lipopolysaccharide has a unique structure, they can use serum types to identify what kind of O-specific polysaccharide is. And so these are the core is the same, lipid A is the same. So only this part is different. Okay, and so we'll come back to the impact of this lipopolysaccharide later. And so that's where the gram-negative and gram-positive differ in the outer membrane. So I'd like to now cover a little bit on a few unique structures on the bacteria. So we often say when you look at a bacteria cell, there's not much structure you can see. And one is the wait, what's this? And one is the flagella. And the flagella determines cell motility. And when we think about motility, this is what the flagella do. And there are three different types of flagellas in general. Like this is, so this is Petrotrichoides. This is more common. It has flagella all over the body. And, and this is mono. You have one flagella here. And this is polar trichoides, and what they call loafer trichoides versus Petri trichoides. And so you have lots of flagella in one end. And the flagella function by rotating around. And here I like to highlight a few things, and is where does the energy come from? It uses proton gradient. Remember the proton gradient we talked about so much? And the proton gradient is the driving force rotating this flagella. And it's also very interesting that the hook part, it's bended. And then the flagella itself is kind of like a bended. Almost like a cork screw. Remember, you open the wine bottle, you use a cork, right? You turn and it just goes in, and then you turn the other way, it comes out, right? Think about using that. And as an example, and the flagella structure itself is well studied, and it's being assembled in the middle. And so it grew in the middle, the filaments in the middle. And the filaments is just one protein. The caps are complex, and the hooks are complex. Pieces are all complex. So how does then this rotating thing work? And then drives the bacteria move forward. And so you have the one direction is counterclockwise. So this is the counterclockwise rotation. And notice this is where you find this figure, okay? And so at the end of the class, the first thing you do is you go through my slides and you find every, you label all the figures, and you go to every section with the figures, and that's where you find them easily. So it's hard to describe one way or the other one, rotating direction, so we use counterclockwise or clockwise as a direction. So in one direction, in the counterclockwise direction, and the flagella bundles together. Doesn't matter whether they are petrotrichoides or loafer trichoides at the end. They bundle together and then pushes the bacteria forward. But when the rotating direction is reversed, then what's happened is that this is pulling the bacteria this direction, this is pulling the bacteria that direction, and this is going that direction. Now, what's the net result? Well, the bacteria doesn't know where to go, so it tumbles. If you look at the videos, and you can actually see the bacteria just don't know what to do. They're being pulled. Just imagine you have 5 friends pulling you all 5 directions. Which direction you go? Well, no, you don't go anywhere. Well, assuming that all your friends have equal strength, right? It doesn't work if you have one friend is 6 feet tall and have double the strength. Then you get pulled by that guy. Okay, and and that's true with this one flagella too, is that you can have one flagella going that direction and reverse and going the other direction. Okay, so the net result is that when bacteria is in a liquid, it will just go one direction for 5 seconds, and then tumble for 5 seconds, and then run for another 5 seconds. So the key here is that the direction, the bacteria somehow just keeps the direction spinning in one direction for 5 seconds, and then it will spin in the other direction for equal amount of time. So the bacteria, Flagella spins around one direction for some time, then spins in same direction in another direction for the same amount of time. So the net result is that the bacteria doesn't go anywhere; it just floats around, and because when it comes out of the spinning, it doesn't know which direction it's going, right? And so it goes this direction, it goes here, and then reverse, and then comes back, and then here, and then go, right? Net result doesn't go anywhere. However, when there's a food, we all want food. Bacteria wants food. And what do they do? Well, they can control that if they're running towards food, they will run it longer. If they run away from food, they will run it shorter. Okay, see the direction here? This is shorter time. And they say, we're not running away from food. We're running towards food. Okay, so that's what the flagella can do. And if you put the, you can test these, and you put them in, put a solution, put a solution with things in there, and then into the bacterial cell suspension, and after a few minutes, you can come back and see. And you can see, well, bacteria voted, right? And you would say they love what's in here. And they hate what's in here. Smells terrible. But maybe there's nothing. I don't care. It doesn't matter. You see, the density outside and inside is different. So that's where you can see. All right. So that's how you can see. You see perturb, right? And the attractant, they will increase, repellent will decrease, and control just being flat. Right? This is control, nothing there, and repellent has no increase. They accidentally got in, and they get trapped. There are other motility mobile mechanism. It's very, now what does this remind you? Like a tread on a tank, right? And so exactly like this. Okay, so this is a relative movement. So one is going the other direction, and so so that resulted in the cells being moving. And so then the bacteria can glide on solid surface. Now, this is a very, very unique mechanism, belongs to one particular bacteria. Does E. coli does E. coli have the ability to glide through on the solid surface? No, absolutely not. It doesn't have that ability. So that's cellular motility. And the next, I want to just mention inclusion bodies. And so we'll come back to talk about this in the future. And the bacteria, when you look inside the cell, they don't have much structure, but some bacteria, remind you, some bacteria have these things. Let me see. Let me highlight. Some bacteria, okay? So that needs to be clear. So some bacteria has what's called the PHB, some has gas vesicle, some has endosport. What's PHB? Well, PHB is this. hydroxybutyrate. So bacteria uses carbon source, right? Eats carbon for energy. So if they have excess carbon, do they just don't take it, or they will store it? Obviously, they will store it. They will keep. They will never give it stop taking food. All right. You give them more food, they will just keep eating, and then store them. And so these are polyhydroxybutyrate. And if you think about biodegradable plastic, this is the first example of biodegradable plastic. And there's other gas vesicles. So here's an example of gas vesicles in cyanobacteria. We know cyanobacteria does green and photosynthesis, right? Well, if you do photosynthesis, you're going to need to be closer to the surface of the water, right? So how do you stay afloat? Well, we swim. What about cyanobacteria? Or we can get a plastic container with airs, right? That keeps us afloat. Well, that's what the bacteria does. They produce a gas vesicle. They produce, so this is across, you see, these are the parallel pictures, but if it's a cross section, you can actually see this cross section of cylinders. And these are side view of the cylinders. And so the cylinder looks like this. And it's made by two proteins. The bacteria only needs to have two proteins, two genes, to make this gas vesicle. And so that allows them to flow in water to get to the surface. All right. And why? Well, they need more light to do photosynthesis. And another very well known, we saw pictures of them, is the endosport. Endosport is made by vegetative cells and turns into spoilating cells. And what we call this is the mother cell, and then releases mature spores. Now, the process of making endosports is actually pretty complex. And this is the last slide. And so this is the vegetative cell cycle, goes from one cell to two cells and just keeps cycling. But when conditions bad, meaning when nutrient runs out, they run out of nutrient, they don't want to die, well, what do? Well, they start making endospore. They use all the energy that they reserve. They separate and then the key here is this step. They first separate, and then this layer, they get invaginated into here. And that's how you generate this stage. And so topologically, this cell, so this still is a cell wall. And this cell wall is being made stronger and stronger by the mother cell. And the mother cell then produces a code. And eventually the mother cell will program itself to die and release the endospore. And that's the process of how endospore is formed. Okay. So, so these are three examples of inclusion bodies that the bacteria can have. Okay. And so today we've talked about cell morphology and the microscopes that we use to view them, and we've spent a lot of time talking about the modifications, and then we talked about the cell membrane, cell wall, and cell motility, and the inclusion body. Okay, So one way to think about this is to use this particular slide as the review outline, and to think about, close the book, say, okay, what did we talk about in cell morphology? What did we talk about in microscope? What do we, you go through the question, and then you go back and ask, go back to the textbook and your notes to see what we discussed. Obviously, the first thing you do is to have we have a lot of blank space on each of the sides. You need to fill up about what we talked about, what we discussed in the lecture, and then you read more, and then you go back and forth and read more textbook and read the slides and to make sure, and then you use this as a guideline to check and see what we talked about. And one of the things I wanted to add is that you typically get a friend to hear what you're talking about. But the first thing you do is actually you don't need a classmate from your class. Just grab your best friend and then try to tell your best friend what we talked about, cell biology. See, can you hear it? All your friends or your siblings or your parents, if they have time.